I'm going to talk about the work that's going on in my lab. So my name is Paul Katz. I'm in the biology department. I'm also the director of the Initiative on Neurosciences. And if you're not getting my monthly newsletter, please sign up and you'll hear about what's going on in neuroscience on this campus. And it's IONS. I don't know, just Google IONS in UMass. There's some other site there. So neuroscience makes use of many different species for research. And you know about mice and zebrafinch and amphibians and zebrafish and Drosophila, C. elegans. You probably don't hear much about polychaetes, but there are people out there that do polychaete nervous system. And then molluscs. And of course, giant brains evolved only twice on this planet, in molluscs and invertebrates. So it's kind of important to understand, if you want to understand how the nervous system really gets to higher level of cognition, it's good to have another example of it on the planet. And the organization of the molluscan nervous system is not the same as the vertebrate nervous system. So if we want to understand what the general principles are, instead of just the family resemblances, we should look elsewhere. And gastropods are the small brain cousins of cephalopods, but they have large individually identifiable neurons. So I've focused on that for my whole career. So gastropods also have a long history in neurosciences. So many of you might be familiar with aplysia, which is Eric Kandel won the Nobel Prize for studies of learning and memory using aplysia. There's older work, LEMAX, having to do with olfaction. So some of the original studies of understanding how olfaction was encoded and the role of oscillations was done by Alan Gelpert and Dave Tank in LIMAX. And the LINNEA has also been an important system for studying learning and memory. Sorry. This is my second talk today on a different, actually, I'm a lecturer. So I'll try to keep up with myself. In my lab, I studied these different species for the last 30 years, more than 30 years. And the work on Tritonia, for example, was started in the 1960s by Dennis Willows. But none of these systems are what you would consider model organisms for modern neuroscience research because they lack the tools. But I want to talk to you first and give you some idea about the focus on the individually identifiable neurons, and then talk about how we can make these systems into more general species that can be used in the lab. In fact, I want to point out that all of these animals are wild caught. I had to hire people to go out, trawl or dive to collect them, ship them across country and across international boundaries at great expense. So it's not an easy task to develop that infrastructure. So as I said, the focus in gastropod neuroscience has been on the large individually identifiable neurons. So this neuron called SI1 is found, this is a cell body. Look at the scale bar. That's a neuronal cell body. It has a ipsilaterally projecting axon. It's surrounded by serotonergic neurons, but it itself is not serotonergic. It uses this neuropeptide for merfamid. So these are characteristics, these three characteristics, the location, the axon morphology, and the phenotype of the neurotransmitter are enough to identify this one neuron in the brain, one on each side of the brain, and only one neuron that fits these characteristics. So you can find it from animal to animal to animal. And then you can also find it from species to species to species. So here's another nudibranch called Dendronotus. It has an ipsilaterally projecting, a cell with an ipsilaterally projecting axon. It's a large cell body near the serotonin neurons, but it contains merfamid. So again, the same characteristics can define the neuron across species. We can also then look, because these neurons have very large soma, we can put electrodes into them, we can record their activity, we can stimulate them. So this is an intracellular microelectrode recording. These are two microelectrode recordings, one from the left and one from the right cell. And if you inject current to cause it to fire action potentials, you get this hyperpolarization of the membrane potential. So it becomes more negative, and then it fires an action potential there. If you do that same thing in Dendronotus, you see the opposite effect, that action potentials cause a depolarization of the membrane potential. That's because there's electrical coupling, gap junctions between these neurons in Dendronotus, but mutual inhibition between the same neurons in Melody. So the same neurons can have different connectivity. And we could do this over with other neurons that we find in the brain. So the ones, the twos, the threes. and they all have different connectivity across the midline. I mean, across these two different species. So the same neurons, they're individually identifiable, but they're not connected the same way. And when we look at the overall activity, these are the central pattern generators for these swimming behaviors. So the swimming behaviors are the same, they're just left-right body flexions. They're actually homologous in that all of the species within this clade of nudibranchs all swim like this. So it's a homologous behavior, the neurons are homologous, but the neural circuits have diverged. And that's a principle that can only be figured out by looking across species to see that. And you can only do that if you have these large individually identifiable neurons. So I wanted to understand how this developed. But we couldn't because these animals couldn't be raised in the lab. So I just want to first put up there conclusions from 3 decades worth of work. Some of you people weren't born yet. I started. So first of all, as I told you, the neurons, the synaptic connectivity, and the behavior are separate levels of biological organization with their own evolutionary histories. That neural circuits can contain cryptic individual variation. So we found that because we knew the identity of the neurons, we could tell if those 2 neurons had the same connection strengths and so forth across species, and we found that they didn't. But mostly it doesn't make a difference for the function of the animal until it's stressed to a point. And then you get differential effects of injury, for example. We also found that rhythmic activity is an emergent property of the circuit that's based on its connectivity and its properties. So that swimming behavior, that left-right alternation, was not produced by any of the cells in isolation. It required the whole circuit working together. And the initiation of a behavior is not a matter of just exciting the circuit. The circuit itself as a functional unit does not exist until neuromodulation, which I'm not going to talk about here, reconfigures it. So this is, I just talked this morning in this thing that David Moorman's organizing called Neuroscience, and we went into detail, talked about this for an hour. But I'll just skip over it now. Because the focus of this time was to talk about what we do next. And the problem with this classical approach in gastropods is it simply does not scale to a brain, even a brain of only 10,000 neurons. We can't go cell by cell or pairs to figure out the wiring diagram of a brain. It ignores the neurons that lack obvious markers. So we can know the neurons that we know, but what did Donald Rumsfeld said? There's the known knowns and the known unknowns, but there's not the unknown unknowns. It also ignores the large number of small neurons. We have the small number of large neurons, but it ignores the large number of small neurons. And it ignores the organization of the inside of the ganglia, the neuropil. And the species that have been used have long generation times, so you can't do developmental studies on them. So Ben Matthews and Leslie Vossel wrote this paper about how to turn an organism into a model organism in 10 easy steps. So first, choose an interesting or important organism. Check. Learn how to rear or work with the organism in the lab. That's the next step. So, and then assemble its genome and profile its gene expression. So these are the 3 steps that I'm going to show you today. So the species that we're going to talk about is the nudibranch Berghia stephanieae. So I joined UMass in 2017 and changed from working on all those other species to focusing on this species, Berghia. So I had an undergraduate in my lab at Georgia State, Amira Hurst, who's shown here. She wanted to do some development. I said, well, we can't raise these labs. We'll find a species that we can raise in the lab. And so she went online and found a species that's being sold to aquarium enthusiasts. So if you get a infestation of the sea anemone called Exaptasia pallida in your aquarium, it takes over. And Berghia is a specialist for eating them. So it can clean out your aquarium, and then once the anemones are gone, the Berghia die because they only will eat that. They won't eat anything else. So it's a very handy system. And then we could raise them in the lab. So they go from eggs to adult and then back again in under 2 months. And you can vary that with temperature. When they start off, they're laid in these egg whorls, these strands. These are individual embryos there. And then they grow up into larvae. This is a typical gastropod developmental stage. It's called a veliger larva. But unlike many other species, they stay confined to the egg capsule. In other species, they are free and they float around the ocean and swim. And so that would be a pain in the butt to try to keep with, from getting away. So now they stay in the egg capsule until they metamorphose and emerge as a slug. And then they grow up in a few weeks to 6 weeks, and they become adults, and they can start laying eggs again. This is our Berghia breeding facility. And I want to, you can just keep them in a tub of water on your shelf, and they will lay eggs and so forth. I want to contrast this with the Aplysia californica breeding facility. It's a multi-million-dollar national resource that requires a whole staff of people to raise the animals. And they also have to worry about their juveniles. They keep them in rotating containers, and they have to feed them microalgae, which they grow. And then they have to feed the adults their algae, which they also have a farm for. So this is our Aptasia farm, which is another tank in the lab. And again, these are cnidarians. These are not plants. So these guys are eating plants. These guys are eating sea anemones. And the reason that they feed on, actually, I should just show you that they feed on the sea anemones because they're sequestering the stinging cells, the nematocysts, and they're putting them in these serrata. Any questions so far? Yeah, interact with them. I got a mic and a PowerPoint.
Do you breed the anemones as well?
Yes. In fact, Akiko Usama, something her name, is a biology faculty member, and she's been working on that for her class. So yes, you don't have to breed them even. You can just cut them in half and they grow two. So they do both sexual and asexual reproduction, but you can get them to spawn as well as in the lab. But the problem that we're having actually is keeping up with the hunger of the Berghia. So we can breed the anemones, but we have to breed them in masses. And so that actually has been the choke point right now. Not breeding the Berghia, but breeding the food. And we're just gearing up. One of the things that we thought of doing, and we're trying out, is to outsource. So we're paying students $1 an anemone for every one over an inch that they give back to us. So we'll give them a starter kit and say, go wild. And then when they come back with a bunch of anemone, we pay them for that. So we just started this. We're gonna see if it works with local students, and we're gonna go to high schools, and we'll see, it might work. That way we're not having to keep these masses of tanks. Basically, these services that you order from online they have a whole, complex full of anemone tanks, never mind the Berghia. Other questions, thoughts? All right, so this is, like the octopus, Berghia grows continuously. So it starts off small, this is 500 microns, it gets up to about 2 centimeters as an adult, and its brain is right there between the eyes. So we can actually, eventually what we want to be able to do is do optical recording from the nervous system in a behaving animal, because we can see the brain while it's moving around. And the brain grows. So the cerebral ganglion starts at about 500 neurons and goes to about 5,000. The rhinophore ganglion starts with 0 at metamorphosis and then grows to 9,000 in 6 weeks. So there's an addition of neurons, and this is actually the subject of my NIH grant, is how does the nervous system function while increasing in size 100-fold over a certain period of time. So we're looking at how the neurons get incorporated into circuits. And because these neurons are individually identifiable, we can just look at the cell body position and gene expression, in this case, the gene for pigment dispersing factor, which is a neuropeptide, and see that there's these 3 neurons, and then we can find them in the J2, the J4, and the adult state. But we also see that there's an additional pair of neurons that come on before the J4, and another pair that come on between J4 and adult. So as the brain is growing, it's adding neurons, and those neurons are individually identifiable, and we can look across specimens, because these are fixed specimens, it's not live imaging, and follow these neurons and see how they're being added to the circuit. We want to get to live imaging eventually so we can watch this happen in real time, but we haven't gotten there yet. Okay, so we chose an interesting organism, we know how to rear it, so now we want to assemble its genome and profile its gene expression. That's the next step. So we published a paper already now on the chromosomal level genome for Berghia. This is in collaboration with Jessica Goodhart, who's now in the American Museum of Natural History, and Deirdre Lyons at the Scripps Institution of Oceanography at UCSD. One of the things, this is a high quality genome, so we now know how big it is. It's got very complete genome. It's, I believe, 18 chromosomes. So it's a chromosomal level sequence. And we found that it contains a high proportion of clade-specific unannotated genes. That is, here's the total number of genes, it's about 25,000, and of those, some are only found in Berghia, some are only found in a subclade that contains Berghia, some are only found in nudibranchs, and some are only found in molluscs. And of these, many are unannotated. So we have no idea what they're doing. So there's a lot of room for biological discovery in here. If most of the genomic work has been done on a small number of organisms that don't include mollusks, then we're going to find genes that haven't been found before, and we don't know what they do. So we've done single-cell RNA sequencing, and we get clear clusters of different things. Here's the brain showing the gene expression for 3 different neuropeptides. And again, just looking at this brain, you can identify these 2 neurons as being unique. These are the only cells in this part of the brain that express the neuropeptide from SCP. There are other SCP neurons in other parts of the brain, but they're unique by just gene expression and location of cell body and size of cell body in this case. You can actually identify that neuron by its DAPI staining because its nucleus is so large.
Maybe said this. This is fish.
This is in situ hybridization chain reaction. It's fluorescence in situ marker, but it's specifically hybridization chain reaction. Other questions?
Are those big neurons polyploid?
Yes. I'll get to that.
Okay, I'll be patient.
We can also look at the neurotransmitters. So this is the glutamate transporter, tryptophan hydroxylase, and choline acetyltransferase, so markers for glutamate, serotonin, and acetylcholine. And we see that we can cover basically almost all of the cells. We also have markers for GABA, histamine. So we now have we didn't have these markers before. Before we did the genomic work, we were relying on antibodies. And now we know exactly what the sequences are for these genes. And so we can label them very specifically. And we can recognize the individuals. So these serotonin cells are actually cells that I worked on for many years. But I can recognize them in this other species because of their location and the fact that they're serotonergic. However, many of the genes, as I mentioned before, are unannotated. So here are these clusters. The clusters are color-coded up here. And the genes that are most differentially expressed across the clusters are unannotated, which are all these yellow bars. So the things that differentiate the clusters from each other the most, we know nothing about. And this gene, for example, that we just give the number 1707, it's only found in nudibranchs, not just Berghia. We see it in other nudibranchs, and it's only expressed in the serotonin neurons that I know so well. So we know nothing about what this gene is. Now, an interesting question about that, the gene sequences might not be homologous to sequences that we know, but the 3D structure might be. So one question that could be looked at is, if you take the predicted structure and you fold it, does it look like things that we know? Has there been drift of sequence while preservation of function? And that's been seen in other genes, but I don't know that literature very well. We also have transcription factors that mark divisions of the brain. The interesting thing about molluscs, one of the interesting things about molluscs is that they're not segmented. So you know about the Hox genes and how they code for the location and so forth, but this is not a segmented organism. And yet the Hox genes and other types of transcription factors show an anterior-posterior expression. So we see this 6,6 is expressed anteriorly, but not in these nuclei and these ganglia down here. We also see that transcription factors that are known to have roles in cell identity and specifying neuronal identity differ from the standard model. So in Drosophila and C. elegans, the transcription factor called UNC4 labels cholinergic neurons. And so it's been thought that it is a cell specifier for cholinergic neurons. But in Berghia, it's found only in the serotonin neurons and not in the cholinergic neurons. So that falsifies the concept, the general principle that UNC4 specifies cholinergic neurons and suggests that maybe it's specifying other aspects of the cell type. So most of these serotonin neurons are in fact efferent, as are the cholinergic neurons in Drosophila and C. elegans. All right, so how to turn an organism. So we assembled the genome, profiled its gene expression, and now develop a method to introduce genetic material into the organism. This is where we are right now. So we haven't developed that method yet. We're working on it. We're trying to figure out, we can inject the embryos and cause them to transform, or cause gene expression anyway, but it generally kills the embryo. So we're trying to find a method for doing that, doing electroporation and other things. That's where we stand on that. It's not the end of the talk. So I want to talk about connectomics. So this is an early version of the fruit fly, the Drosophila connectome. This has been one of the major transformational events in neuroscience, I think, is having the full brain connectome of the fruit fly. That has changed how research is done in these animals, because now you have the wiring diagram of the brain, you have every single neuron, and then they have reporter lines that allow them to insert genes into any cell that they want using this the split GAL4, no, I forgot which system they're using. Anyway, they can put, it's the GAL4 system, and they use a way to have lines that allow them to insert genes into any particular cell type. They have a whole library of them. And it's revolutionized how people work on Drosophila, and I want to be able to do this in a mollusk now. So first off is creating a connectome through volume electron microscopy. So I've been collaborating with Jeff Lichtman, and he's one of the earlier pioneers in connectomics. What basically the tissue is put on this thing called an automated ultramicrotome. The sections are collected on tape. That tape is then put down onto a silicon wafer and then imaged in this case, a multi-beam electron microscope. This is the first brain that we started. So these are the rhinophore ganglia, the cerebral ganglia, and then the pedal ganglia. This is before sectioning. So this is a micro CT image of the brain that we then section. This is a side view of not the same brain, but it gives you the general view. So here's the animal's head. These are the rhinophores. The eye, the brain sits between the eyes. So here's the eye, here's the brain. And we started to section from the front, and that's where COVID hit. And we had to stop. So we were left with a partial data set. But this is part of that data set. This is half of a rhinophore ganglion that we've assembled. And it's changed my view of how these systems work, because this is the large number of small neurons that are in the rhinophore ganglion that we know nothing about, and now we're starting to learn about them. So Harshita Sant is a postdoc in my lab, and she's been coordinating this. I should say, so during the pandemic, she was going to join my lab in March of 2020 from India, and then that didn't happen. And so we figured out how she could control the electron microscope at Harvard from her home in India. And so she was able to do the scanning of the sections from India. And then she arrived here, and she's been coordinating work between our lab and Jeff's lab, and doing this with a team of undergraduates. So I've had 10 to 12 undergraduates in the lab working on this project. And they are hardworking, tracing these cells from section to section. So here's an example of a neuron that Sarah found that has its cell body in the rhinophore ganglion, projects into the cerebral ganglion. That's the end of our data set. That's the frustrating part. It has these dendritic branches. And so Kriti identified the neurons that are presynaptic to that one. So the ones that contact it, and then she's able to trace back. And so now we can start to build these circuits out. What type of neuron is this? Who's before it and who's connected to that? We're doing this manually right now, but we're working with Jeff's lab to get the data into a position where it can be automatically segmented. And that's where most of the field is right now. You do the automatic segmentation and then you proofread those results. Basically, we're going to end up with a position where we can click on a neuron and then click on the cells that are presynaptic to that and work out the whole circuit. There's also a part of the rhinophore ganglion that we knew about already. This is from another undergraduate's project. So she used a small cardioactive peptide antibody and showed that there was this region in the rhinophore ganglion that had these axon terminals. And Sarah found a terminal in that region, not necessarily the same cell, but a cell of a similar type that had lots and lots of synaptic boutons, these bulbous parts that are filled with vesicles. So here's this blue one right here. And it's just chock full of synaptic vesicles. So she segmented all of the boutons around it. She wanted to see if there were other neurons. And what she found is that yes, there are cells that are interacting with each other. And this is kind of striking, that there are Bhutan to Bhutan membrane contacts and no postsynaptic elements. These are all release sites, but there's no obvious receptor site. But this also fits with what's being found right now in the striatum, that there are Bhutan to Bhutan connections that are communicating through non-synaptic means, so the neurotransmitters are being released, and then the receptors are not at standard synapses. She also found that some of these Bhutans weren't connected to neurons at all. So there were these weird, they look like synapses, they look like they were synaptic Bhutans, they're filled with vesicles, but they were hanging out, and when you do the 3D reconstruction, there's no neuron attached to them. But instead, they seem to be being engulfed by glial cells. So we might have been catching pruning of the synaptic terminals in action. In addition to the neurons, any questions so far? Feel free to jump in with anything. Yes.
What is a bouton?
It's a French word for button. And so basically, a neuron has these, so this is the axon comes down, the action potentials come in, and then there are these swellings. And the swellings are where the peptide vesicles are. So those vesicles get released from those swellings. And so that's just called a bouton. It just means it's a swelling where the vesicles are. And it's characteristic of where synapses, where neurotransmitters are released from. Yeah, so please, if there's something that's not clear, let me know. So Alex is another undergraduate in the lab, and he found ultrastructurally identified these cells as glial cells. And then he reconstructed them. So here's a 3D image of glial cells for the first time in molluscs. They look like they could be astrocytes, or they could be microglia, we don't know. So we're gonna correlate this with the gene expression to see what kinds of glial cells they are, because we have 3 clusters of glial cells in our single cell sequencing data. But this is Des Ramirez. He, again, did the work on the single cell sequencing, and he found that in addition to the small number of large neurons, there's also a small number of large glial cells. So these are the giant neurons, and then this is a giant glial cell. We had no idea that they existed. In fact, we thought many of these nuclei were neurons, but in fact, they're glial cells. There's a large number of small glial cells as well, and these are small glial cells that are up in the rhinophore ganglion. But these large identifiable glial cells are something that we're going to look into further. Yeah, Jen.
So to do this and say that these are glial cells, are you using markers from other systems like
No, this is
Drosophila?
apolipoforin is a marker from Drosophila.
Right, so yet you've described how they might be very different from the model systems. So how confident are you applying marker genes?
So lumping and splitting is basically what this comes down to. There's obviously things that are the same across all animals. So sodium channels, you can recognize it as a sodium channel based on its sequence, but there are going to be differences in the types of sodium channels. And I think this is true of many of the things. Yes, the 6,6 is present as a transcription factor, or UNC4 is present as a transcription factor, but it's different in its selectivity of what it's doing. So yes, one always needs to apply these homologies with care. But the marker for a glial cell turned out to label glial cells. It's not a neuron. You can tell from the morphology that that's not a neuron. And anything in a nervous system that's not a neuron is by definition a glial cell. So we have now the next series of specimens that are ready to go for sectioning. So these are juveniles now that are embedded and ready to be sectioned for volume EM. So we're ready to move on that. This is a micro CT image of one of these, and you can see, again, in the embedded tissue, the cerebral ganglion. So we know that the tissue is looking good, and we're just going to start sectioning it and then reconstructing it. And we hope to do some of that at the EM facility here. So we have a Thermo Fisher volume scope that I was a co-PI on the instrumentation grant to get that, but we haven't used it yet, so we're waiting to use it. Almost there. All right, so we want to define what the inside of these cells. We're going to do this with the EM, but we can also do this at the light level. So Cheyenne Tate's another postdoc in the lab. She's been using neuronal tracers. This is neurobiotin, and she applies it to the nerves in the rhinophore that then go into the rhinophore ganglion. And she's then able to see where those axons end up. And so they go to specific places in the cerebral ganglion, as well as into the contralateral rhinophore ganglion. So right away, what this tells us is that there's bilateral connections between the neurons in the rhinophore, not the brain neurons, but the afferent neurons that are coming into the brain, and they're already making connections in the two rhinophore ganglia. The other surprising thing was that there's a photoreceptor in the eye. So this is the eyeball. This is a photoreceptor, and it has an axon that is getting filled by putting tracer in the nose. So there's a primary sensory neuron connection between the eye and the rhinophore, which is a primarily olfactory organ. And that helps explain an observation from 1978. So Dan Alcon recorded from photoreceptors in another nudibranch, Hermesenda, and he showed that they are inhibited when applying an odorant. So that was just kind of left there. And now we see that there is actually a direct connection between the photoreceptors and the olfactory system. Phoenix Quinlan, who just defended her PhD a week and a half, 2 weeks ago, has been studying the visual system, and she found that you put them in an arena with a stripe and they will orient to the stripe, they'll go to the stripe, and they'll straighten out their paths even if they don't go to the stripe, if there's a visual target. And it turns out they only have 4 photoreceptors in their eye. This was known about nudibranchs in general, but now we have the option to show that in fact this is true, there's only 4 photoreceptors. But if you increase, I didn't show a slide. Anyway, if you increase the gain on this, you see that the opsin is actually can be traced out into the rhinophore. So you can see those axons that contain the opsin going out to the rhinophore. Questions? Yes.
Does this suggest they might be able to like see smells?
So, yes. We also have unified perceptions of things. So neuroscience historically studies systems. We study the visual system, we study the olfactory system, we talk about them as if they're separate. But as you know, we experience it as one. So, right, like when you're watching your screen on your computer and the sound is coming into a headphone or a speaker that's on the side of the computer, you experience it as one. There's this binding that goes on. And I think that in these animals, the binding is happening really early. So it's been known since Dan Alcon's work, for example, that you can do conditional classical conditioning of a nudibranch, where you shine a light, which has no effect, and then you rotate the slug and it clamps down. And if you pair the light with the rotation, the slug will eventually learn to clamp down in response to the light, which it didn't do before. And that plasticity occurs in the photoreceptor, which is normally it's thought that plasticity should happen in the brain. So my conclusion, like if you were to ask me, I think that it's all one nervous system and that you can have these things that are happening all over the place.
Just in nudibranchs or?
So octopuses also have autonomous arms. They have more neurons in their brains than mice do, and they have 3 times as many neurons in their arms. And this, I think, comes from, I don't remember if I have that slide. I do have the slide coming up. It comes from the way that our nervous system and their nervous systems differ in early development. So let me get back to that. If I don't have it, I'll answer you. So Cheyenne then was looking at bilateral integration, and she showed that using different color tracers, that there's this region here in the brain where you have strong contralateral representation overlapping with ipsilateral representation. She also looked at synapsin, which is a marker of synapses. So the red is now the glial cells, and they're marking the borders of the synaptic regions. We can differentiate different types of synaptic regions. So this is all synapses, and these are synapses that contain this marker of plasticity called protein kinase AC alpha. And this is a marker of a learning region in the insect brain, the mushroom body. So here's a very specific neuropil structure that has a ring in it that is staining for this, and the next door neuropil does not have that. Here's this neuropil here, which is where those synaptic boutons were, and it also is expressing this marker, but other synapses in that same ganglion are not. So there's something going on here in the organization of these neuropils that we're still discovering. All right, so now to development. This is work that was, the last time anybody looked at the development of gastropods was 1984. Michelle Jacob is at TOPS. We're actually scheduled a Zoom meeting with her next week, or maybe it's this week. I gotta look at my calendar. And we're gonna talk about our work with this and where she ended up in 84. So the way that the nervous system works is that there are these ectodermal placodes and the cells migrate in postmitotically to form the ganglia. This is the embryological development. This is really different than a vertebrate where you have neurulation and then the nervous system develops from the neural tube. The central nervous system develops from the neural tube, and then the peripheral nervous system develops from neural crest, which is between the neural tube and the ectoderm. And so all of the peripheral nervous system is coming from neural crest, which migrates away. All of the central nervous system is coming from inside the tube. That's a fundamental difference between vertebrates and invertebrates, and specifically with molluscs, where the neuroectoderm stays external. Well, we don't know what happens as the animal's growing. So now we have evidence that there are migrating neurons in a post-embryonic animal. So these are cells that are found between the ganglia and the skin that express ELAB and another unannotated neural marker. So they clearly have a neural phenotype, but they're not in the ganglion. And they're in between the ganglion and the periphery. So we're looking to see if we can, again, we want to be able to do live imaging to follow these cells and see where they end up. We found that there's proliferating cells all over the body. So this is the oral tentacle, and these are the neurons. So ELAB is another neuronal marker, a gene that is expressed only in neurons. And we see that the proliferating cells in the neurons are intermingled in the periphery. We also looked at markers. Phosphohistone 3 is a marker of mitosis. We see mitotic cells in the brain as well as in the periphery, but these could be other types of cells. They're not necessarily neurons. So we're looking further into that. And then in early juveniles, there's very little difference in the cell size. So here's the cerebral ganglion, and here's the rhinophore, and you can see that the neurons in the brain and in the periphery are about the same size. It's not till later that they start to grow massive. So when we look at the massive neurons, though, we see that the full adult giant neurons are containing markers that indicates DNA replication. So this gets to the endoreplication. I forgot who asked that. Did you ask that, Larry?
Asked about polyploid.
Polyploid. So, but how you get polyploid is you replicate your genes. And so here you have the giant neurons that are showing markers for DNA replication, which should be only in mitotic cells, but these guys are post-mitotic. They're fully formed neurons, and they're replicating their DNA. So the question is, why are they doing that? And there's been explanations put forward, like, why do we have jump? Is that something we should worry about?
I don't think so.
We're not.
Somebody with a cart.
Oh, I thought it was a bomber. Right, so the giant neurons, why are they giant? And one possibility is that they're giant so they can produce more gene product. They replicate their genome and then they can produce more gene product. All right, so as I mentioned, ganglionic neurons migrate in from proliferative zones, and then they increase in size and replicate their DNA. But we don't know where the peripheral neurons are coming from. Are they coming from special proliferative zones or anywhere? Are they coming from the same proliferative zones but different stem cells, or even the same stem cells? So these are issues that we're looking at in my lab. I also want to mention that we're studying the behavior of these animals. So I talked about the visual behavior already, but Kate Otter is a grad student in the lab, is studying their feeding behavior. So they feed on these sea anemones, as I mentioned, and they're getting stung. So here the animal got stung and it's moving away, and it's making these choices. It's coming back, you see how it's getting stung and it's not liking it, but it goes back in anyway because it feels so good, I guess. But it's a fair fight. This is a sea anemone that's now eating a sea slug. So the sea slugs do feed on these giant anemones, but if they happen to get caught by the mouth, then it's not good for them. Cheyenne's been looking at their reproductive behavior. She has actually, I'll say that in a second, but these are two slugs that are in the process of mating. So they have to recognize each other as conspecifics. So what you're going to see is that they're going to come around, they're going to face each other, and the first thing that they're going to do is, the next thing they're going to do is go mouth to mouth. So here the two mouths are touching, and they put their tentacles out to the side, and then they line up. So each of these animals is a hermaphrodite. They're both male and female, and they exchange sperm one at the same time. So they have a single gonadopore, which acts to both deliver sperm and receive sperm. And you can actually see it in a second. You can watch the packets go from one animal to the other. I'll try to point to it. Ready? It's going to happen over here. And there's one, there's one, there's one. Can you see it? There's one. Are you able to see that on the screen? I don't know. Oops, next slide. So she's identified the phases. There's kissing, where you're putting their mouths on each other for a while. And then there's transferring sperm, and that's the short act. And then they cuddle for 6 hours. So why do they cuddle for 6 hours? It's to prevent cheating. So just to be clear, nudibranchs kiss and cuddle to prevent cheating. So they recognize each other, and then they mate, and then they hold each other to prevent the other one from reaching back and eating the sperm that they just gave it. So that's the way hermaphrodites would cheat, is that they would just snack on it instead of developing those eggs. And then they lay the eggs. And then Cheyenne has identified the peptide hormone, egg-laying hormone, ELH, that is found in all gastropods, but the sequence has diverged in the nudipleura, so in the nudibranchs and pleurobranchia. So she's identified that, and she's synthesized it and shown that when you inject it into animals, it causes them to lay eggs. She's looked at the expression in the brain, and she's found a cluster that's asymmetric. It's on the right side, which is where the gonads are. She's also seen ELH expression in the periphery. So here's a peptide that's related to egg laying, but it's also doing other things in the brain, and we don't know if it's coordinating behavior across the whole animal at the same time. And this paper just got accepted in Hormones and Behavior, so that's going to be coming out shortly. Phoenix, as I mentioned, is doing the visual navigation. I already talked about that. And then she's graduated, so I've got room for other PhD students coming along now. So in summary then, Berghia is a tractable system for transcriptomic, connectomic, and developmental studies. The individual neurons can be identified and tracked over development as the brain grows. We're working towards developing a full brain connectome, and we have to learn how to express genes in neurons and how to do live imaging of neurons. So I want to thank the members of my lab who actually did the work. This is the lab as of last May. We need a new group picture. And our funding we get from NSF and NIH, and it was the initial collaboration with Jeff Lichtman and Dee Lyons came through a Brain Initiative grant. Thank you.